So for those graphs, you want to make sure that you have all of the information on there. Label, metric units. I need to know what those numbers are. Label, we don't need metric units on this one, but I need a label to show what those units are. Watch your numbering. The numbering has to be consistent. It doesn't matter what numbering patterns you chose. So Logan stole his, and this happens to be counting by tens here and counting by tens there. That's fine. You are more than welcome to do that so long as it fits in all of your data. But you don't have to. So I stole Dixie's as well. Dixie counted by fives, 5, 10, 15, 20, 25, 30. She counted by fives on both. That's fine as well, but we could have even done one of each. One side could have been counting by tens, one side could have been counting by fives, and that's fine too. It doesn't matter what counting pattern you choose, so long as it fits all the data, and it must be consistent. So a lot of people on their graphs put down 0, 10, 30, 60, 120, 180. And Dixie did this too, and so you might be saying, hey, well, why didn't you put big red X next to that one? Because on most of the ones that I talked to you guys about, so you had 10 here, and then you had 30, and then you had 60. So what you're saying is that those two squares is worth 10, and then those two squares are worth 20, and then those two squares are worth 30, and that's not a consistent counting pattern. So the only numbers that she put on here were the numbers from the lab. However, she spaced them out, so it's showing that each box is worth 5 seconds. So it's okay just to put those numbers on there. But once again, you have to have a consistent counting pattern. If it's easier just to write the whole thing out, write the whole thing out just so long as you have a consistent counting pattern. I'm going to roll with Dixie's on this one just because it's spread out just a little bit more. So, you have your graph in front of you. Our control on Dixie's graph is the purple line, it looks like. Somewhat purple. That printer doesn't do too well on there. Okay, so I want to look at the control and compare it to when we increased enzyme concentration. We increased enzyme concentration, we doubled it, right? Sound familiar? Because we had 2 people working instead of 1. So we doubled enzyme concentration. Do you see what happened to the line when we doubled enzyme concentration? So here, we've got a nice little steady, and we could find rate for this as well. We have a nice steady rate, and then what does this flat portion mean? No more reaction. The reaction basically stopped. We have 0 additional broken toothpicks. So we went to here, and then we didn't have any more that we broke because we used up all of our substrate. So on this one, same thing. What does the flat line mean? No more reaction. We used up all the toothpicks. We used up all the substrate. So, did that happen at the same time for both of those situations? When we doubled the concentration of the enzyme, when did we run out of substrate sooner? At 60 seconds. And it took us another whole minute in order to run out of it with our baseline. So for enzyme concentration, when we increased the enzyme concentration, our rate went from this to this. So we can use rate on this as well. But I like to compare all of the rates on the line of best fit later on, because then we take this part out. So, steeper slope. We were going through toothpicks faster. We ran out of toothpicks faster. Steeper slope. It went through the substrate faster. In that experiment, raise your hand if you were an enzyme. Did we use up our enzymes and they were gone and we didn't have enzymes anymore? No. We had our enzymes throughout the entire situation. Now, in our body, usually enzymes are not used up. They're always there. However, if we don't need the enzyme anymore, our body has ways to break them down and recycle it. I mean, why have it sitting around if we don't need it? So, technically speaking, we can clean that out, but we don't use up the enzymes in this process. We use up the substrate because the substrate's gone, but we don't use up the enzymes. All right, so this was our control. This is when we increase substrate. This is when we decrease substrate. So this is when it runs out, and we're not worried about running out. I want to look at this part right here when we still have enzyme concentration. Those lines for that rate, is there a big difference in them? For these three right here, I mean, we've got a little bit on this one. Is this our second one? Oh, that's our control, right? That's our control. Decrease in substrate, increase. Oh, okay. Perfect. Didn't even realize how perfect this was. This looks like the rate's a little bit slower, right? Here's the thing. Our enzymes were humans, and the first thing they went over there and they're like, oh, I've got to break toothpicks. Okay, I'm breaking toothpicks. Then when they realized that we get to the point and we're trying to run out, then all of a sudden it was a race to break the toothpicks the fastest between the three of them. Right? You changed how you broke them, right? Because I saw that, and I was like, oh, I needed to point that out, and I completely forgot about it until I saw that graph. So here's the thing. I'm totally taking that little bit of difference out of the rate because of the fact that my enzymes actually changed how that was another independent variable. And maybe at the very beginning I need to change that and say, hey, you need to do it as fast as you can. That way you're doing it as fast as you can the very first time. And then I would bet that those lines are going to be even closer together. So when we change the amount of substrate, did that really have an effect on the rate of reaction? It's all relatively the same right here because it's the enzyme, and the enzyme can only work so fast to break a toothpick. They're going as fast as they can. They can only do it so fast. And so it doesn't matter if there's a ton of substrate or a little bit of substrate. They're going to still do it at the exact same rate, or relatively same rate. The difference is, when do we run out of substrate? So when we had double the substrate, we never ran out in that 3 minutes that we worked. When we had 10 grams or 5 grams, and this is 5 grams compared to 10 grams, we ran out a lot sooner when there was less substrate than when we had the 10 grams. Does that make sense? But if I double the amount of enzymes, if I increase the enzymes, it makes sense. I've got more people snapping toothpicks, so they are going to do that faster. Questions about that graph? All right. AP Bio Lab Notebook. Now you got to bear with me just a little bit because I need to redo this notebook too. Because every year I'm like, oh, this is good to add. Oh, this is good to add. And then I mess things up. So notes are going to be a little bit squidgy. What's the last thing that we had in there? Oh, okay. Biodiversity was the last one. And then your cytochrome worksheet, make sure that you go ahead and get that in there as well, if you haven't already, where you did your phylogenetic tree. That is actually that's a blast lab, so that will count too. Like if somebody wants to look at your notebook and say, hey, did you do labs in AP Bio and give you lab credit for it? Then you want to have as many of those in there as possible. So the cytochrome C is where we had those papers and you counted differences in DNA. And then I gave you that back. So make sure you take that in somewhere too. So you might want to save a page for that. Okay, let's do this first. My notes are going to be kind of jumping around a little bit. So let's do enzymes at work first. So I've got 2 pages side by side. This is an AP Bio Lab notebook. Enzymes overlap HBS and AP Bio. For HBS, we talk about it in terms of digestion. For AP Bio, we talk about it in terms of the process of how they work and what effects environment has on it, what effects concentration has on it too, but we leave that in HBS. All right, so catabolic reaction, anabolic reaction. Summarize for me. If I say catabolic reaction, essentially what does that mean? What's going on? Okay, catabolism, we're breaking it down. Anabolism, okay, and taking this and this, and we're bonding it together. So we're building it up. Catabolic reaction, typically we're breaking down polymers, breaking down into our basic macromolecules, or breaking down macromolecules into their monomers. Anabolic, we're building things. All right, I got a little slip of paper on the front table. Has a diagram that looks a little bit like this. And in fact, let me go through this first, and then I'll have you come up and get it. Because you won't be listening to my melodious voice if you come get it. Tape it in now. All right, so perfect example of catabolism and anabolism. Maltose is a disaccharide. Maltose is a disaccharide. It's a carbohydrate. It's a sugar that's made of 2 glucose molecules. If I want to break this down, then we're going to use the process of hydrolysis. And what does hydrolysis mean? What am I doing? Yes? With water. Hydrolysis. So hydrolysis, the hydro part, we're adding water in order to break these apart. So I go from disaccharides to monosaccharides. Anabolism of glucose, I can create maltose. So I take 2 glucose molecules, and I have a condensation reaction. So in order to have a condensation reaction, what do I need to do? Take water out. Do you remember the other name for a condensation reaction? Dehydration synthesis. So I'm taking water out. So here, the chemical reaction is building it up. We're bonding it together. Here, the chemical reaction is breaking things apart. Okay, go ahead and grab that real quick, and then you'll tape it in under those 2 terms. Just a good example of those reactions and how they act in nature, in real life, if you will. So we did that. Go ahead and tape that in real quick. Yours should go below it. Mine's above it. Like I said, my notes are all over the place. I have to redo this particular set of notes. Just tape it in below anabolic reaction, catabolic reaction. All right, so now I suggest that you put the next pieces of information on the next page. So I suggest that this goes on the right-hand side page because we're talking about a different substrate, and so that might be a little bit confusing. So our cells produce H2O2. Do you know what H2O2 stands for? So H2O2 is a naturally produced waste product. Our cells produce H2O2 naturally as a waste product of reactions. It's toxic, and it needs to be catabolized. So what do we need to do to H2O2? We need to break it down. Our cells produce H2O2, that's hydrogen peroxide, naturally as a waste product of reactions. It is toxic and needs to be catabolized. And if you don't know what H2O2 stands for, make sure you make yourself a note. H2O2 is hydrogen peroxide. So hydrogen peroxide is toxic to us. Say that one more time. Sure. Yes. So you probably have a bottle of that at home, and that bottle is probably brown. nice and dark. This reaction, hold on to that thought. Hold on. I got to do something else before I get to that thought. But come back to that and remind me about the cuts and scrapes thing. I'm getting ahead of myself. So excited. So, have you heard the joke? Guy walks into the bar, tells the bartender, I'll take a glass of H2O. Bartender pours him a glass. There you go. Guy sitting next to him says, I'll take a glass of H2O too. Bartender says, okay. Pours him a glass. Guy drinks it, and he dies. Because he gave him H2O too. Gave him a glass of hydrogen peroxide. So he died. Oh, that's good. That's quality science. So we naturally produce hydrogen peroxide, and we need to get rid of it. This reaction of breaking down hydrogen peroxide, what do you think? Just looking at that chemical equation, what do you think that we can break hydrogen peroxide into? Because what's the chemical formula for water? H2O. So I see H2O in there. So when hydrogen peroxide breaks down, it breaks down into H2O and O2. Do you know what O2 is? Oxygen, and specifically, this is the kind of oxygen that our bodies can utilize. So environmental science especially, we talk about ozone. You know what ozone is? Have you heard of ozone? Is ozone a good thing? Technically, it's a good thing in the stratosphere, but down here, it's bad. So we have incomplete combustion that produces ozone, and it's a toxin. It'll kill you. Chemical formula for ozone is O3. What do you know about ozone based on its chemical formula? Sure, okay. Give me a little more information. There's 3 oxygens that make it up. So that seems like, well, more oxygen, well, that should be better for us, right? But the way that they're bonded, our body can't break them apart, and so it ends up being a toxin and it'll kill you. O2, the way that that molecule is bonded, our body can break it apart and utilize the oxygen. So O3 kills you if you breathe it. O2 kills you if you don't breathe it. H2O kills you if you don't drink it. H2O2 kills you if you drink it. Just these minute little changes. We are so technical and so complicated. So we break down hydrogen peroxide, and the reason why it's in that bottle, that brown bottle, is because if it were in a clear bottle, if it was sitting out on a table, and even in the brown bottle too, it's going to naturally, spontaneously break down over time. Any idea what kind of reaction this is classified as? Because it occurs spontaneously, naturally, essentially by itself. Exergonic. So if we were to take this reaction, if we were to take hydrogen peroxide and just sit it on the table, eventually the liquid that's in the glass is going to be water and the oxygen will be in the atmosphere. Now, I don't trust it enough to drink it after a little while, but there are probably tests that you can use to run through and it'll prove that. The problem with that is it takes a long time. It's not like you set out, poof, and it happens. So in our body, if we're producing hydrogen peroxide and it's a toxin, do we want toxins laying around? No, we don't. Severin does, and that's why Severin died before the end of Biomed. But we don't want toxins laying around in our body. We've got to get rid of them. So we need to speed up the reaction. Even though it's exergonic, even though it's going to happen spontaneously, we need to speed that up a little bit. So what are we going to use to speed up that reaction? An enzyme, because an enzyme is a, with a C, ends with ist. Catalyst. It's a catalyst, and catalysts are things that help to control reactions, especially speed them up. So using an enzyme, which is a catalyst, helps to speed that process up and make it go really quickly. So what temperature do you think that the liquid would have if we were to speed up that reaction? Do you think that the glass would feel hot or feel cold? Okay, what kind of reaction did you just tell me it was? The catalyst is the molecule, but what kind of reaction? Logan just told me exergonic, and I agree with that. What do exergonic reactions do? Do they absorb heat or give it off? Do they absorb energy or give it off? Exergonic exit out endergonic, endocrine gland, the hormone goes right into your blood. So exergonic reactions, it's going to give off heat. It's going to give off energy. So we speed that stuff up. You can feel it, you can feel that it's feeling hot. So if you have a cut and you pour hydrogen peroxide on it, that reaction speeds up. Because if you don't have a cut and you put it on there, then it's kind of like water. But if you have a cut, especially if it's exposed to blood or bodily fluids, and you put it on there, then all of a sudden it starts bubbling really quick, right? That's because we have an enzyme in our blood that helps to catalyze the reaction to break down hydrogen peroxide, which is good. We need that enzyme in our blood because we're producing H2O2 as a byproduct. So we need an enzyme inside of us to catalyze that reaction and break it down. The enzyme happens to be in our blood and our bodily fluids. So when it's bubbling up like that, it's turning into water and oxygen, which conveniently enough, the water helps to wash stuff out and the oxygen helps to kill anaerobic bacteria. So bacteria that don't like oxygen, and if they come in contact with oxygen, it kills them. So if you get like a nail or something jabbed down deep in, anaerobic bacteria can get in there and they're like, oh no, oxygen, nice and warm and moist, and they multiply and divide. Tetanus is one like that. It gives you sepsis, gives you infections in your blood and stuff like that. But a little bit of hydrogen peroxide gets that oxygen down in there, and they're like, no. And so it can kill them even though they're not exposed to oxygen that's in the atmosphere. So the enzyme that's in our blood is speeding up that reaction, is catalyzing that reaction. All right, we've got that part. What do I want next? Oh, okay, let's do this part next. All right, so go ahead and add this next. So our immune cells, no, this keeps going with your H2O2 stuff. Our immune cells purposely make hydrogen peroxide. The little devils. They do it to kill proteins, bacteria. They do it to kill pathogens. We are so complicated and amazing. If you go into anything biomed related, when you get to college, there are so many different areas that you could go into. Find something that you're like, oh, that's cool. And then there's probably a whole subject on it and a whole profession on it. Our immune cells produce hydrogen peroxide purposely to kill pathogens like proteins and bacteria. And then we have an enzyme in our bodies that can break hydrogen peroxide down into its waste products, water and oxygen gas, which it's easy for us to get rid of water. You're going to use the oxygen gas. All right, so next, let's write down the equation for that process. Two hydrogen peroxide molecules will break down into two water molecules and one O2 molecule, one oxygen molecule. Hydrogen peroxide in this situation is the substrate. The water and the oxygen are the products. This reaction occurs naturally, but it takes time. You need an enzyme to speed up the reaction. So the enzyme of importance here is catalase. Catalase is the enzyme that breaks down hydrogen peroxide. Catalase is not a substrate. It's not a product. It's an enzyme. It's just a molecule, protein, that speeds up the rate of reaction, and it's not used up in the reaction. So one of the reasons why that hydrogen peroxide is in such a dark bottle is because sunlight, the environment, that's going to speed up the reaction too. The dark, cold environment, cooler environment, if you put that in, your hydrogen peroxide is going to last longer. And one last term with these notes, free energy, never hurts to have a definition down there. Free energy is also known as activation energy. It's the energy within that environment that's available to do that work, to get that reaction going. Catalase, that enzyme, that catalyst, helps to reduce the amount of free energy you need to get things going. All right, so once you've got that, turn. Let's do the next 2 pages side by side and actually put your lab stuff. So this is investigation number 13, enzyme action. Our overarching, our big old question, what effect do environmental factors have on enzyme action? So we've actually already touched on what kind of things can have an effect on enzyme action. In our HBS course file, we listed things like substrate concentration and enzyme concentration. And we just looked at that. We just looked at how substrate and enzyme concentration affects enzymes' rate of reaction. Enzyme concentration sped up the rate of reaction. Substrate concentration didn't have an effect on the rate, but you ran out of substrate at different times. The other thing that we talked about in HBS having an effect on enzyme rate of reaction was the inhibitors, and we'll look at that when we finish up the graphs that deal with your rates, all 6 of those rates. Then the last thing that we talked about were environmental factors like pH and temperature and stuff like that. So we're actually going to focus on environmental factors. Since we've already looked at those others, we're going to focus on environmental factors in this activity. So first of all, what does the H with the little O mean? Null hypothesis. So what's going to be a null hypothesis for this experiment? Not just no effect, but what? Yes, but not. There's no statistically significant effect of environmental factors on enzyme action. So go ahead and write your null hypothesis. So what does null hypothesis mean? And specifically, not just no difference, but, all right, and then not just no difference, because let's say 1 is 1 and 1 is 2. There's no statistically significant difference in the effect of environmental factors on enzyme action. All right, so what's H with an A? So alternate hypothesis, that's the actual hypothesis. So you have to write a hypothesis. The environmental factors that we are going to affect will be, we'll have freezing temperature, boiling temperature, high salinity, and I think I can do both pHs. I have to check, but I think. So we have high pH, low pH. So we're going to have cold temperatures, freezing temperatures, boiling temperatures, hot temperatures. Probably won't be quite boiling because that's a little dangerous. Salinity, do you know what salinity means? And somebody said it? So specifically, this solute will be salt. So saline solution that has to do with salt. So high salinity, high pH, low pH. High pH is basic, low pH is acidic. So you need to choose one. Which environmental factor has the most effect on enzyme action? And write a hypothesis. Make it a somewhat explanatory hypothesis. So you choose one of these, blank will have the most effect on enzyme action because, and tell me why did you choose that one? We are, but that's not part of our hypothesis. I mean, we are going to be using hydrogen peroxide and catalase. But we're keeping things a little bit. Well, I guess that may have an effect on your because. Yes, so the substrate that we're using is hydrogen peroxide. The enzyme that we're using is catalase. so we're going to put a graph on the left-hand side. So just skip to the next page, and this is information that we need in order to collect data. So investigation number 13, environmental factor. Depending on who's here, we'll put you into groups. I'll assign you an environmental factor. We've got to have some for the afternoon as well. So you'll just write down which environmental factor that you're collecting data for. I'll do that first, and I'll explain it. I put this on the next right-hand side page. It just needs to be somewhere around what investigation 13. But it's easier to write on the right-hand side pages, I have found. All right, so you're going to be using hydrogen peroxide and catalase, and catalase is the enzyme that breaks down hydrogen peroxide. Catalase is an enzyme. You can't tell it, whoa. As soon as it comes in contact with the substrate, it's going to start reacting. So one of the things, no matter which situation that you're at, and it'll make more sense tomorrow when you see your setup and we go over that, no matter which situation you're at, as soon as you add the enzyme, there's going to be a stopper in order to collect your gas, and then you're going to start timing and collecting data. Bam, 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 bam. It has to happen fast, or else you're going to lose data. You're going to lose some of the gas, some of the product from it. So you have to be careful with that, and you have to make sure that you work really quick. So you'll have 1 milliliter of catalase. Hang on, just listen for just a second. My melodious voice, and I'll let you write down. Everyone will have 10 milliliters of hydrogen peroxide. Everyone will have 50 milliliters of a solution. It kind of depends on which station you'll be at. Some of you will just have distilled water, DiH2O. Some of you will have a pH solution or a salt solution. But everybody is going to have 50 mils of a solution. It just depends on which factor you're testing. And the catalase, you'll have it set aside. Don't add it to the other stuff until you are, bam, ready to go. That should be, you got everything ready, you're good to go. Put it in there, and then the experiment is off. Some of the situations may not seem that way, but trust me, it is. All right, so go ahead and add that information. We will need to have a control setup, and the control setup is going to be at room temperature since the others are freezing and boiling, essentially. I mean, probably not exact. But, pH of 6, which is relatively neutral. That's usually what our tap water runs at. We'll use distilled water, so no buffer solution in it to change the pH, no sodium chloride in it to change the salinity, and 20 degrees Celsius is about room temperature. So that goes with this, sorry. Room temperature is about 20 degrees Celsius, pH, distilled water, since we're testing temperature, pH, and salinity. Yep. 0% NaCl, 0% sodium chloride. All right, then you need a data table. Got metric units. Gotta have metric units. so what's on tap for tomorrow? And does anybody else know if they're going to be gone tomorrow or Friday? Friday? Okay. So what's on tap for tomorrow is set this up, talk about this, and then run the lab. It takes a little bit of time, and then of course you're going to analyze the data for it. I expect you to have about 30 minutes while we wait for the other buses to come in. So those of you that are on late buses, you need to understand that at that time that we're waiting on you guys, we're working on the digestive system stuff. So that's something that you either take care of outside of class or do it on the bus ride over here or something along those lines. That needs to be finished tomorrow because on Friday, then we're going to talk about that, and that's going to be a class grade. I'm going to ask you a question, and you have to be able to answer it. So remember, we need to collaborate, and remember, Braxton's got that number 4 questions, so make sure you touch base with him. That morning period while we're waiting, if Abby brought us the eyes, if you are interested in dissecting one of those eyes, then you've got to kind of come in early or be ready to go when you get here at 8 o'clock, 8:10, whatever. Then we can do a couple of those eyes, and then a couple of them are going to be for the afternoon. So that's the max of 2 people. Ideally, you would come in a little bit early before class in order to get that taken care of, and then we can look at that. But then that would be early tomorrow, and then if nobody's able to do that, then I'll just do it so that we can look at, especially I want to look at the lens and the cornea, because since they're not preserved, they should give us another look at what's going on. The cornea might start being cloudy because at this point, I'll give that in a minute. At this point, you know, the eyes, you know, the cells start to break down, so it might be cloudy at this point. Okay, and then tomorrow we will do a review, so you need to make sure that you go over endocrine system, enzyme stuff in your HBS course file. Free response question is due Friday when you walk in, so those of you that are going to be gone need to turn it in tomorrow. And then finish up that other graph and get that into me as soon as possible, and we'll get that taken care of. All right? I will see you tomorrow.